Hey guys, welcome back. Hope you had a good weekend. So we're going to take a practice test. We're going to a practice quiz because we're planning on taking a quiz over the first 4 chapters on Wednesday. Tomorrow, Tuesday, we're going to have a short review. We're actually going to finish up this unit after chapter 5, which is a really, really important chapter 5 over z-scores and standard deviation. And bell curves, very, very important chapter we're about to start. We're going to finish that all and take the test over the first unit September 30th. So if you've fallen behind on homework assignments, all of that Unit 1 homework from chapters 1 through 5 is all going to be due on the 30th of September. So if you have fallen behind, that's kind of your deadline to get everything caught up. I won't take any more of this homework after that point because once we move on to Unit 2, bivariate data, I want to make sure we're all moving on together. So this video is going to be really, really long. I'm just going to warn you because the very first part of the video, actually I might break it down into 2 spots. I'll probably break it down into 2 spots. How about that? I'm going to make a review for the entire unit. If you are good and you don't think you need a review, or if you want to watch the review at like 2 times the speed, even though I'm going to talk fast anyway, I'm going to review over all the highlights of all the slides and not all the slides, but pretty much everything that we've done up to this point. So it's going to be pretty epic. I am excited. I'm not going to go into as much detail, so it's just going to be a general overview review. Okay, and then I'll probably break it out into a separate video, which will actually be the solutions to the practice test. So once you've taken your practice test, you'll have the PDF just like a normal homework assignment. I want you guys to mark it up and to work through the practice test just like it's a homework assignment, and you can upload it and submit it again just like a homework assignment. And then you can watch the solution video and go through the solutions with me to see if you got it right. So make sure that you got it right. So let's go ahead and do our review really quick without any further ado. So first question is, what is data? So data is gathered all around us. It's got what is some information that's also recorded along with its context. So we need to know what it is, where it was, where we got the information, what time we got the information. We need to know what kind of a variable it is, what kind of units in it, if it does have units or anything like that. So for instance, we have a data table from Amazon. We got all these different variables here, but without its context and knowing what each one of these variables stands for, it's really confusing and it's not that helpful. And especially if this was if we didn't even have the context of Amazon. I mean, if we see Amazon here, even that kind of helps us figure out what's going on. But if it was just data, raw data without its context, it wouldn't be very helpful. So next thing is a data table. So data table is going to have the data with the variables labeled, which are usually in columns, and the actual individual case is going to be a row here. So each row is each case, which might be each purchase. In this case, it would be we have variables and cases. So the variable the data table tells us what it is. For instance, name, state, country, price, and the case itself tells us who. So in this case, the horizontal rows. All right, the context, the who, what, when, where, why, and how. So the who is the population of the sample. So what are we trying to draw conclusions on? We're talking about all U.S. registered voters. The sample is who we actually sampled and gathered information from. The what is going to be the variables. What questions did we ask them? What responses did we get back? What did we measure? Stuff like that. The where is the location of the population of the sample. So if we're talking about the U.S., of all the U.S. voters, that would be all the U.S. Our sample, if we only talk to Oklahoma voters, then that would be obviously a sample that's only Oklahoma, which would not be representative. You'd have to sample from the whole population, right? When is what the time the data was collected, and the how is the method for collecting the data, which is very important that you're transparent about your method so that we can judge whether or not we think that it's going to be representative. Gathering data, we can do it through surveys or studies. So surveys, just like you think, is when you send out a survey to people where you ask them questions or you track them down or interview them or something like that. A study is going to be like an observational study, which is where you observe things in nature, kind of like right here. So we're observing it. We're not playing an active role or manipulating or messing with these albino rhinos or whatever it is that we're observing. We're just observing trends in nature. Experiments, on the other hand, this is how we prove a causal relationship, is when we actively delegate or we actively randomly assign treatment groups. So you get the placebo and you get the active drug. But of course, we don't tell them who gets which. It takes an experiment to be able to prove a causal relationship. Observations and studies, just trends that we notice. We might be able to prove there's an association between something, but which is another way of saying it's not independent, but we can't actually prove that there's a causal relationship unless we have this random assignment of treatment groups. And also surveys, usually for percentages. That's where we get anyway, that's where we get confidence intervals and stuff like that. Like we thinking we're between 40 to 45% of Americans believe in proposition X, Y, Z, whatever. All right. Respondent, someone who answers the survey. Experimental unit is whoever who's participating in the experiment. If it's a human, we usually call them a subject or participant, so that, it's not like a lab rat or something, which sounds really mean. And then record is the information about an individual that we collect from a database. So sometimes we gather data from records, which is not the same as a survey or study or an experiment. It's just something that's already happened and we're gathering the data from it. All right. Oh, that's interesting. Population is sample. So the population is the big group that we're trying to draw conclusions on. The sample is a smaller group that we randomly sampled in order to hopefully get a representation of the entire population. So for instance, of all U.S. voters, that would be registered voters is our population. The sample is going to be the people who responded to our poll, or the people that we found, more importantly, to respond to our poll. Sample and population, so no big deal. We have 3 different types of variables: identifier, quantitative, and categorical. Identifier variables are categorical variables that record a unique value for each case. So for instance, like an order number, that's an identifier variable. If we're talking about a person, we could use like a serial number, not serial number, I'm sorry. For a person, we could use their social security number. That would be an identifier variable to identify an individual based on the number. You could also use maybe like a student ID number. If we're talking about Pump Stage or something like that, that would also be an identifier variable. Categorical variables are variables that we sort into buckets. So like yes or no, or high, medium, low, stuff like that. Those are categorical variables. Favorite color, ice cream type or something like that, favorite ice cream, those would be categories. Quantitative variables have units with them, usually numbers, and we have to have some sort of units that's associated with it, and you know what new units are. A data table is a table where, of course, it's sorted out by the variables that we're asking for, and each one of them is going to be each case. So for instance, all the classes or the sex or the age or whether or not they survived, all of that's in a data table here. If we just looked at all 2,000 or some odd people that were on board the Titanic, that would be a ridiculously large table. So instead we want to look at a frequency table, which frequency just means it's telling us the counts. So how many people were first class, second class, third class, group class. So this is frequency table by class, which is nice. Another version of the frequency table is a relative frequency table. Anytime you see the word relative, it means it's a percentage. So instead of looking at the counts, we take the total and we divide each one of the counts by the total to get the percentage to figure out what the breakup, the percentage breakdown or distribution of class would be. You know what I'm trying to say. Another thing is a bar chart. It's got spaces in between it. That tells us that there's no natural order between like small, medium, large, or anything like that. Unlike a histogram, which is not going to have any spaces, which tells us that it's broken down into bins or categories. This is categorical data, but it's not. Bins break down quantitative data into specific categories. So like a range from 0 to 2, and then 2 to 4, and then 4 to 5, 4 to 6 or something like that. But here, the bar chart, the actual order we put the bar charts doesn't really matter, even though it might feel like there's a natural order like first, second, third, and then of course crew would be the last one, because we're talking about position of where you're sleeping, right? So first class is close to the deck, second, third, crew, of course, is going to have the cramped quarters on the very bottom, and they have to climb the stairs further and stuff like that to get to the deck where everyone wants to hang out. Also, relative bar chart. Again, the relative word means that it's going to be percentage, so it's exactly the same, except our y-axis here is going to be written in percentages instead of written in counts or frequencies. So sometimes relative bar charts are kind of helpful. Difference between a pie chart and a bar chart. Pie charts are when we're comparing parts to the entire whole. So if you want to see which one was the biggest or something like that, you can also see quick estimates for a half, a third, fourth, quarter, 16th, stuff like that, which you naturally would be able to break down. Like for instance, this is a half, that's a quarter, that's an eighth, and that would be a 16th, right? You'd also be able to see naturally what a third looked like, which is something that looks something similar to that one. So anyway, kind of cool. If you want more precise values where each of these are listed in percentages or something like that, for a relative frequency bar chart, then you would actually want to look at a bar chart instead. But sometimes we label pie charts with the percentages on all of them, which is like the best of both worlds. But if you want to be able to compare different values to each other, to other parts instead of to the whole, like which one was the smallest one, well, it's kind of hard to tell which one's the smallest between the blue or the red over here. But when we graph them out like this, oh, it's pretty clear. This one's obviously smaller, right? So more precise values, like that question here, we're comparing to other parts, we use bar charts. Comparing it to the whole, we use a pie chart. This is fun. Hope you guys are having fun. Ooh, I feel like I need to drink some Mountain Dew. All right, so area principle. So I'm just going to show you 3 here. The area should be proportional to its actual value. So whenever we're looking at this, it doesn't actually tell the story because population density is not by area density. So here it looks like usually, the red areas are going to be bigger than the blue areas, but because of the difference between the density, population density of people who live on the coast, it doesn't quite tell you the exact story. So if we remorph and distorted by electoral votes or by population, then the U.S. would look something like that, which is kind of funky. And then it tells a story of why the blue candidate won that year. I don't even remember what year it was. Anyway, clearly it was 08 or 2012. I don't remember. Anyway, it was from the book. All right, area principle. This is nice and pretty. Everyone likes to look at three-dimensional graphs, but it violates the area principle because we're not only growing by length, we're also growing by height, which means that this crew, which is 3 times the length of the second class, relative, about 3 times, is actually going to be 9 times as big as an area because it's also 3 times as tall as this one here as well. So even though three-dimensional graphs will look nice, they're not very valid. It is the same information as we see over here, right? So that is true if you're only looking at lengths, but unfortunately our eyes are drawn to the area. So here, see how they all have the same vertical area, okay, vertical length. Over here, it's not the same, okay. So that's why it's no bueno. So good to know. Area principle, also three-dimensional graphs. This should not even be an option because there's no time when this is ever appropriate. So I know it looks good and Microsoft Excel will produce these for you. Apple's, whatever they call it, will do the same thing. But you should never, ever, ever do that because you have this front surface on the parts that are in the front, but you don't have that where you can see it in the back ones. Like none of it at all for the crew. So even though the crew is the largest group here, it looks like third class is bigger than that, doesn't it? Yeah, that's what I thought. So you do not use this at all. In fact, I'm going to cross it out. No good. Okay, moving on. Contingency tables are going to be breaking down. So you're looking at 2 different variables and you're sorting people into across the board for 2 different variables. So for instance, did you survive or not, and what class were you in? So let's say you were first class survivor, you would go in this category right here, okay, in this contingency table. Were you a non-surviving crew member? And then you go in this one over here. Once we have those contingency tables, typically they will have marginal distributions. The marginal distribution is going to be whenever you collapse one of those 2 variables. So if we only want to look at class and not survival or death, then this would be the collapsed version. So instead of sorting it out by class, now we're going to collapse it down. So we're only looking at one variable at a time, which is the class. Same thing with marginal distribution for survival. We don't care what class, just if you survive or not. We're taking all 4 of those and we're collapsing it down into just one variable. It's 2 variables. We're going down to 1 variable. Did you survive or not? That's called marginal distribution. Think about margins of your paper. It's like the sides that go around the edge. Conditional distributions are percentages. Percentages of what? Well, to be able to find out what your percentages of, look for the 100% on the graph. So in this case, the bottom right-hand corner is 100%, which means it's a total of the overall. So what percent of people were first-class survivors out of the whole boat? 9.2. What percent were crew that did not survive? 30.6. Over on this one, we see the percentages on the right side. So we're looking at, of all the people who survived, what was the breakdown of the class distribution. So this is class distribution by survival, and this is class distribution by not survival. Okay, kind of cool. We can display those class distribution by survivals in a pie chart. And if it was, we would expect the same percent survival rates. In other words, no. Anyway, whenever we graph those out, both the alive and the dead, they should have the same areas. If they don't, then that means that there's an association between the 2. Kind of cool. All right, so another way of looking at this is the survival rate. So this is distribution of class by survival, distribution of survival by class. So they switch because on this one, we're distributing the class by whether or not you survive. So if you survive, what was the distribution of your classes? And then this is the distribution of survival by the class. So if you were in first class, 100%, what's percent of your survival? So these are what we call survival rates. These are the death rates, which is really morbid. Notice that you really only have to graph 1 because those 2 are complements of each other, meaning they add up to 100. So if we know that the survival rate goes down as you're further below the deck and your class goes down, then we know that your death rates go up because they have to, right? Those 2 are tied together. So it's actually a little bit ambiguous to graph both of those. So also we would expect all these to be the same across the board if they were independent, meaning that if I knew that you were in first class, it does not affect your survival rate. And of course they were not independent. because of their location to the boats. Same thing between these two. We would expect these to be exactly the same area, and since they're not, that tells us that the crew members and the third class were way overrepresented when it came to the people who died, and they were way underrepresented when it came to people who survived. And these first class people here way overrepresented the amount of people who, proportion of people who survived, where they underrepresented the people who died. So that's what's going on there. All right, independence versus association. So distribution of survival by class. So these are the death rates, which is what we graphed here, very morbid. We would expect all classes, regardless of like how many people were in it, when we're looking at the conditional distributions, we would expect all of the proportions of death rates to all be the same, and they're not. So that tells us there's definitely an association between the two. Segmented bar charts gives us the exact same information that we get from a pie chart. So here I graphed out the alive and death percentages, right? So we've got the distribution of class by survival. The exact same information over here. See the 29%, that's this part here, and the 45.2%, that percentage there, is the exact same thing we have over here. Okay, so what's nice about putting it as a segmented bar chart is that you can actually measure out the percentages a little bit easier, but even better if you just label them on a pie chart. I prefer that because then you don't have to go from a weird spot to another weird spot and subtract or do anything like that. But again, you see the areas are not equal here, the areas are not equal here. See how those green ones are way different. The pink ones are about the same. Yeah, that was about the same. But you see some other differences between the other classes. So that means that they were non-independent. All right, so different ways to display quantitative data. So here we have quantitative data. So it's going to be fit into bin sizes. This is a stem and leaf plot. We use that for smaller data sets up to 45. Anything bigger than 45 typically, just as a general rule, we use a histogram. What's a plus side of a stem and leaf plot is it shows the actual data. Another version of a stem and leaf plot, which is similar to it, is a dot plot. Dot plots don't actually show the actual data, the numbers themselves. It just shows dots for each category, so it's not as good. So anytime you use a dot plot, there's no reason why you wouldn't use a stem and leaf plot because it's got a perk to it, and they basically do the same thing. All right, so small data sets, we can use stem and leaf plots, and it's kind of nice to know the actual distribution of the data. If we have larger data sets, then we want to use a histogram. Notice that the histogram and the stem and leaf plot give you the exact same overall general shape or the distribution of the shape of the distribution of whatever this was. I think it was number of states visited. But if we have even larger data sets or we're comparing multiple histograms to each other, then we want to use a summary like a box plot. Box plot is going to summarize the information from the stem and leaf plot or the histogram by giving us the quartiles or the 5-number summary, which is the minimum, first quartile, median, third quartile, maximum, and that's also going to tell us if we have any outliers if it's a modified box plot, which is really nice. So those are 3 different ways we can graph out and quantitative data. The other one is going to be a scatter plot or a time plot, and I'll show that in a little bit. This is fun. Flying through all this stuff. All right, so describing with histograms, which we usually use, we want to describe the shapes of the distribution. We're looking at the mode. So unimodal means it has one clear, obvious mode or most frequent bin or category, or like if it was a general shape of a mountain, it has one peak. Bimodal, which typically happens where we're looking at difference between subgroups, usually male and female, like such as average height. If I graphed out the average heights of all of my students in statistics, there's going to be 2 clear modes. One's going to be the mode for the males and one's going to be the mode for the females. Kind of cool. Multimodal, that happens for lots of different reasons, but it means there's multiple modes. Uniform means it's almost exactly uniform or the same across the board, like rolling a dice. Another way with shape, when we have unimodal data, we also sometimes want to describe it as symmetric. If it is unimodal and symmetric, meaning if we fold it over, it matches up almost exactly, just fairly well. It just means it's not skewed, in other words, right? Then we can actually use a bell curve, which we'll learn about next week, which is very, very powerful, and we can do z-scores, and everyone likes that stuff. Another thing is that if it's not symmetrical, it means that it is skewed. So then the question is it skewed to the left or is it skewed to the right? So to find that out, we just simply follow the tail. Data that's skewed to the left means that there's usually a reason there's a hard right bound over here, and it's still more variation that's allowing to happen on the low side. Like for instance, most people that have cardiac issues like heart attacks, they're going to be older, right, in their golden years, but there's not going to be many people over 100 that are still having heart attacks because no one's really living over 100. It is possible, but even more rare to have a heart attack even at the young age of 25 or something like that. So there's definitely more range or motion that can be happening on the left end, left tail instead of the right tail, and that's why it's skewed to left. On the right side, it's looking at CEO data or the amount of money that they spend or something like that. Mostly it's going to be here. You can't spend less than $0, but it always can get more and more expensive depending on what they have to do to fix it. All right, unusual features like outliers and gaps. Gaps happen when we have gaps between two groups of data. This is not considered a gap because this is an outlier. It's not really a group of data like a split in two. You know, we got this group and this group over here. So this would be considered an outlier. Gaps also don't happen when there's just one missing bin or one empty bin. That's just because it's a small sample size. As we sample larger, eventually we'd find somebody that fit in that category there. So this is not considered a gap because there's not multiple spaces in between it. Also, it's not really a group of data. I guess you could call that a group. So these are probably not going to be outliers either. So there's really nothing going on over there. This is considered a gap because it's between two groups of data and there's multiple bins that are empty. This is probably going to be an outlier because if we do our interquartile range times 1.5, it's probably going to reach out about here, and it's not going to be over there. So it'll fail our left fence test. And we calculate those mathematically. How do we do that? Good question. Here's a problem from the homework. So we look at 1.5 times the interquartile range. So we measure the interquartile range, which is going to be Q3 minus Q1 multiplied by 1.5. So it's about 1.5 times the length. We add it to the third quartile and we subtract it from the first quartile. So on this one, looking for the upper fence or right fence, this would be the right fence. Anything beyond that point is going to be considered an outlier. And then we put our whisker going to the next point that's below within the fence. It would not be considered an outlier. Same thing here, 1.5 times the interquartile range. Add it to the third quartile, subtract it from the first quartile. That'll tell us that any point beyond this point right here, which there was one right there, it's going to be considered an outlier. So we show it in the modified box plot. By the way, a box plot is also sometimes called a box and whisker plot. The box is going to be the inner quartile, and the whiskers are going to be the fourth quartile and the first quartile, or in other words, the point going to the minimum and the point going to the maximum. When we want to know what typical behavior is, we basically just look at the box, the interquartile range. That's the most important part. The whiskers just tell us, does it, like if it's skewed, right? Do we have a longer right whisker or do we have a longer left whisker? Which both of these have longer right whiskers, which means that the data is going to be skewed to the right, even without even knowing about the outliers. All right, so ways to measure the center, we either use the median or the mean. The mean is going to be the balancing point, which actually takes into account further points are. The further they are from the center, the more they're going to impact the balancing point, right? So whenever we're dealing with a median, the outliers, whatever happens at the 1% or the 100%, doesn't really impact what's going on at the middle 50% or the 50 percentile. Okay, we will have equal areas on both sides, and it doesn't matter how far spread out each side is, it's still going to be equal area. So if we have unimodal and symmetric data, we can use the mean, but for any other types of data, bimodal, multimodal, or skewed data, unimodal and skewed, we are going to use the median and the interquartile range because it's not impacted by outliers or skewed data. So in this case, because we have an outlier and the data is skewed to the right, we cannot use the mean. We have to use the median because it's not unimodal and symmetric. We will use mean and standard deviation after we take the test, and it's really important and impactful when we do have data that's unimodal and symmetric because we can use bell curves. All right, on this one we use the mean or standard deviation, again skewed to the right, so we have to use the median again, can't use mean and standard deviation. We can also measure the spread. The spread is basically measuring the amount of variation, or how far the data is varying from the center, or is it very consistent, or is it all spread out? In other words, we never use the range because the range is the difference between the max and the min. It is entirely impacted by outliers. For instance, if we remove this one outlier here, then the range would be significantly smaller. So never use the range. We only use interquartile range and the standard deviation. Interquartile range, basically quartiles break the data into quarters, like so 4 quarters. So here's our first quartile is going to be here. Second quartile is going to be there, which is also the median. Third quartile is the 75 percentile, and then the fourth quartile, of course, is going to be our maximum, which is going to be the 100 percentile. So it breaks it down into quarters. Hey, so look, quarters now. Hopefully you can remember what that means. The interquartile range means the middle quarters, which are the middle 2 quarters, which is going to be from the third quartile to the first quartile. We just subtract that to get the interquartile range. The 5-number summary is basically what we graph out when we graph out the box plot, and it's going to give us our quartiles here, and to find the interquartile range, we just take Q3 minus Q1, which in this case would be 1, right there. Kind of cool. All right, ways that we can describe the spread. If it is unimodal and symmetric, we can use a standard deviation. Remember, we calculated the deviations, which is how far the data was from the average, from the mean. and then we squared it so that we wouldn't have positive and negative values. If we took the sums without squaring it, it would always add up to 0 because they're going to balance each other out. So we have to square it, and then when we're done squaring it, we add it together, divide it by the n-1. That'll tell us the total variation or the variance in the distribution of the data. So how much variation we have. But variation is measured in the units of the data squared, and we don't want the units squared, we want in the actual units themselves. So we take the square root of the variance to get it back to the standard deviation, which is going to be the average deviation in the units of the actual data, whatever we were measuring. So whether it's in feet or inches or pounds or something like that. Instead of pounds squared, it's going to be just pounds, which is standard deviation, which is the average deviation for all the values of the points. So kind of cool. And we calculated that by hand. Remember, that was so much fun. It took like 5 minutes. All right, and the last thing, comparing distributions. So we talked about our graphing the wind, right? So this is going to be a time plot, and it's actually, I forget the actual word for it because it's the lines are connecting it, so you can actually see as it fluctuates. But anyway, these are the daily values. As we average them together, we can graph all the daily averages for the whole year, which would look like this in the histogram. We can clearly see some of our outliers, but this is summarizing what the whole year was like. It's not as nice, doesn't really tell the story of what's going on in different seasons and stuff. So instead, we could graph it out by seasons like summer and winter, and then we can compare those two if we make sure that the y and the x axes are matching up, so we're not stretching it out either vertically or horizontally. Typically, just looking at them side by side, you can tell the difference, but I like to, because I can, overlap one of them on the other one so that you can clearly see kind of what's going on with this ghost or parent graph or something. I don't know what they call that, but that's what I want to call it. Anyway, you can see what's going on between the difference between those two. They're different spreads. One's obviously skewed more to the right. The other one's more unimodal and symmetric. We've already talked about all this stuff before. Then even better, if we're going to compare multiple distributions, like all 12, like for each month, then we're going to have to use summaries of the data instead of the actual histograms themselves. Best way to summarize quantitative data is going to be with a modified box plot. From the modified box plots, we can see the 5-number summary. If we want to know what a typical day would look like, then we're just looking at the boxes. So ignore the whiskers and look at the green boxes. Yeah, you can see what's going on, right? If you want to know whether or not the data is skewed, you can look at the whiskers easily. So are the whiskers equal length on the right and the left, like we see with July, or are they clearly shorter and longer? So the whiskers are good for telling whether it's skewed. You can also look at the median and where it is with relationship to the box. This one's centered, so looks more symmetrical. This one's slightly lower or closer to the first quartile, so skewed, kind of skewed, you can kind of get the idea right there. Okay, also outliers are plotted out by the points, and whenever we break it down by month, we can see some outliers. This would be a very usual point if it was, I don't know, April, but because it was a few months later in July, this is a very unusual point, very windy day. And then you have some like far extreme outliers over here, which we found out was because of, that crazy weather that they had. This is a scatter plot. In particular, this would be a time plot because our x-axis down here is days of the year or some value of time. So as we go from day 1 all the way to day 365, we plot each one of those points. Notice you don't have the lines connecting it. I think that's a line plot. Whenever we did like the fluctuating, I think it was a line plot. But anyway, this is just a scatter plot because there's no lines connecting them. If there were lines, it would be so busy you wouldn't be able to see the dots. But we can see the general trends. Things calm down, then they get kind of a little windier. What I like to do was to take the scatter plot and overlay it on top of our box plot so you can see how each one of the box plots was collected with that month's worth of data. Kind of dope, if I'm being honest. This is how we calculated standard deviation. So we took the actual values, how far they deviated from the means. We squared the deviations. We took the sum of the deviations, divided them by n-1 because we were dealing with a sample instead of the population, and then we take the square root of it to get back to the standard deviation. That was fun. Ogives don't really tell you the shape like a histogram does, but ogives are very nice if you want to know the specific breakdowns of the percentiles, because the left or the y-axis here, our response variable, is the cumulative percentage. So if you want to know what percentage you scored in for cholesterol, which is not necessarily a good thing to have a high score in, then you can look at your value for cholesterol and then or just trace the value to the left, and that'll tell you your percentile. So ogives don't really tell us anything about the shape of the distribution. Well, they kind of do, but they're hard to read, so don't look at them that way. They're really all about percentiles, so that's what they're used for, and you'll have one. All right, so that was a 30-minute review, and I'm pretty sure a few minutes at the very beginning of that was me just talking and giving you guys some announcements. So anyway, that was pretty much me not really reteaching, but reviewing everything that we've talked about over the last 2 weeks, and I did it in 30 minutes. That was like so crazy. So I'm gonna take a few deep breaths and like chill for a moment, and then I'm gonna record the practice test solutions. So at this point, if you want to take the practice test, go ahead and do that on the PDF, work it out just like a homework problem, and then after you've done that, you can come back and watch the solutions with me, which I will post as a separate video, because otherwise you'll see a 45-minute video or 50-minute video and you're gonna freak out and you're not gonna watch it and you're gonna be mad at me. So I'm gonna post them as 2 separate videos, and I'll see you in a little bit. Bye bye.